Great. Thanks a lot, Dennis. So I'll talk a bit about our work looking into de novo mutations and regulatory elements. So I'm a PhD student here at the Sanger Institute, and this project is a part of a much larger project, which is the Deciphering Developmental Disorders Project. I'll refer to it as DDD occasionally throughout this project. So it's based here at the Sanger Institute, but it's actually recruiting patients from all over the UK and Ireland. And so we're kind of the hub of the hub and spoke model. So there are clinicians across the UK when they have a child that has a developmental disorder, frequently intellectual disabilities, that they can't diagnose through the normal means in the national health system here, they get recruited to this study. So at the moment, we have about 10,000 trios. These are affected children plus 2 parents. And we have clinical phenotyping on all the children. So for example, I've put here about 80% of the cohort has intellectual disability or developmental delay. About 10 or 11% have congenital heart defects. There are also a number of abnormalities, including growth issues. So for all of these 10,000 trios, the data that I've got here is in the first 8,000. We have whole exome sequence data plus about 5 megabase of non-coding elements, which is what I focus on. I'll get into in a second exactly what that 5 megabase of non-coding elements entails. So the first and kind of the basis of all the analysis that I'm doing here is based on a mutation rate model. So ideally, the question we'd like to ask first is, if we look across this set of non-coding elements we've sequenced, do we see more de novo mutations in the affected children than we expect under a null model? So it's important to take sequence context into consideration here. So for example, these 4 big red peaks are CPGs, which we know to be hypermutable. So if you happen to have a sequence that's CPG rich and you don't account for this hypermutability, then you might see more mutations than you expect simply by chance, not because there's any disease association. So in general, I'll be using this model because it's generative. It allows us to take a sequence and simulate a mutation process and say, given a population of a particular size and a sequence with a particular context, how many mutations do we expect to see? And also we can actually just generate example mutations and see how they differ from the data that we observe in our patients. What we also do what I have a sort of natural control because I focus on the children who are already diagnosed. So you can use the already diagnosed children as well as a control, much like you'd use, say, an unaffected sibling in autism. So I'll give you an example from one of my coworkers' recent publications at how this model performs in practice. And it's very well calibrated. If we look at synonymous changes in genes, the number that we observe in about 4,000 families is very close to the number that we'd expect under the model. But in classes of damaging variation like missense mutations or protein truncating variants, we see a large enrichment. And this is reflecting the fact that some proportion of these mutations are causing or contributing to the disorders that the patients have. So you can break these down further and we'll find that protein truncating variants in known developmental disorder genes or in genes that are loss of function intolerant in a healthy population, you'll see even a larger enrichment. But this mutation rate model is pretty good at matching what we expect to see in variation that shouldn't be under purifying selection or be disease associated. So this is another figure from Jeremy's paper. And it's just an example of how successful this trio sequencing approach has been for identifying both known and novel genes that are associated with developmental disorders. But there are actually still about 70% of the children in our cohort that are without a diagnosis from a protein coding variant. So my work has focused primarily on the 70% or so of these patients who are what we call exome negatives. So after looking at the exome, we still don't have a mutation that accounts for the patient's disorder. So the simple question that we want to ask is, first, what proportion of this 70% of missed diagnoses do we think actually realistically might be coming from mutations in the non-coding genome. So to do this, included on the exome capture is 3 sets of regulatory elements. And I'll explain why each of them were included. This was done quite a long time ago, before much was known about regulatory elements and conserved non-coding elements in general, but I think it was actually a really nice study design for my supervisor. So there are about 4,000 elements that are what we call CNEs, conserved non-coding elements. These were selected to be the top 4,000 most highly evolutionarily conserved elements, and no account for any function was taken on those. So it was not known what they were doing or what tissues they were active in if they were regulatory elements. So these cover about 2.5 megabase. The second set were 800 what I'm calling VISTA enhancers. These are enhancers from the VISTA database that have biochemical evidence and in most cases expression data indicating that they were real enhancers in humans and or mice. So these are about 800 elements covering 1 megabase. What I've got on the left-hand side of this plot is just the evolutionary conservation. So these enhancers span from, in some cases, being very highly evolutionary conserved at the top to at the bottom, enhancers that have biochemical activity but are not particularly evolutionarily conserved. And finally, there's a set of 1,200 heart enhancers. These were included in part because when this exome capture was made, there was data suggesting that the set of VISTA enhancers or the set of well-characterized enhancers might have been depleted for heart enhancers. And also about 10% of our cohort has a heart phenotype. So these were included as a way to, in part, boost up what might have been a deficit in well-characterized heart enhancers, and also to check down the line if some of these heart enhancers might be contributing to the heart phenotypes. But as you can see, this set is is not particularly evolutionarily conserved compared to the other sets. So the first thing we wanted to do was characterize these 3 sets of non-coding elements a little more closely. So a lot of you may be familiar with this, the singleton proportion or the mutability adjusted proportion of singletons as a measure of purifying selection. Basically, the way it works, if we look at a class of variation like loss of function mutations that are we suspect to be under purifying selection or damaging in some way, the variant frequency spectrum will be shifted towards the rare end of the spectrum. So we'll see more singleton loss of functions and fewer common ones. And if you look at a synonymous site or a class of variation that's less damaging or under lower purifying selection, the singleton proportion as a proportion of the whole variant frequency spectrum will be lower. So in the EXAC data set and in the paper that was recently published, they extend this to be the mutability adjusted proportion of singletons, because when you have large data sets, CPGs, for instance, become are hypermutable, and so you can actually see a depression in the singleton proportion. Also, if you're looking at a specific sequence context, for instance, splice donor or acceptor sites, you might see differences in the proportion of singletons that are driven by mutability and not necessarily by strength of selection. So I'll use the mutability adjusted proportion of singletons here. As you can see, any elements or any classes of variation that are under stronger purifying selection will have in general higher maps, and synonymous sites are set to be 0 because that's what we use to fit the model. So if we run the mutability adjusted proportion of singletons across all of our sets of non-coding elements, I've also included synonymous sites here as our kind of marker of what is no purifying selection. You can see that the heart enhancers are essentially under no purifying selection, as well as a set of control introns we've included. This is just sequence that's covered by the exome baits that we kind of get for free on the standard exome capture, while the VISTA enhancers and the conserved elements are under purifying selection, although to a lesser degree than coding regions in general, and certainly to a lesser degree than known developmental disorder genes. So I intentionally ignored the kind of variant we're looking at in the genes. So this will include synonymous missense and stop gain in the genes, because that is the same situation that we have in the non-coding elements. We don't really have a way to identify benign versus likely pathogenic variation, but I'll get more into that later. So this is just important for us to get a picture of what might we expect to see in de novo mutations based on what we see in population data. So using the trinucleotide model, this is figure 2 from the paper that we just posted on bioRxiv. If we look at the 4 sets of elements that we originally included, these control introns that you get for free from the exome capture, plus the 3 sets that were specifically targeted, we don't see any enrichment in the heart enhancers or the VISTA enhancers. The red diamond is what we observe, and the black bars are the 95% confidence intervals of the null model I described.
Sorry, can I ask a question?
Sure, please.
So did you see a colocalization of these highly conserved elements close to genes that are also highly constrained? Is there some kind of LD link between the two so that you're capturing some of the signal that leaks out from the highly constrained gene?
You mean with the purifying selection on the previous slide or in the de novos?
No, in the previous slides.
Yes. So I didn't account for that specifically here, the closeness to constrained genes. So what I can say is we exhaustively tried to link these conserved elements to target genes using a number of different approaches, and we didn't really ever find any signal that showed them to be conclusively closer to constrained genes or known developmental disorder genes. But I think you're right that there could be some background selection that's contributing to this. Although a number of these elements are quite distal from the elements, but I think you could do a correction there to account for the proximity to genes that are under constraint.
Okay, thank you.
So if we look at these 4 sets of elements, the only place where we really see any enrichment is in this set of conserved non-coding elements. And all the other ones are very close to what we'd expect under the null model. But as I mentioned earlier, the conserved elements were chosen without respect to their tissue activity or function. So if we split these conserved elements into ones that are likely active in the fetal brain versus inactive in the fetal brain. So the reason we split on the fetal brain is that most of the probands in our project have a developmental or neurodevelopmental disorder. Then we see that that there is in fact some enrichment in the fetal brain active conserved elements, while the ones that are likely to be inactive in the fetal brain, so they're active in some other tissue, liver or heart, etc., don't show any enrichment. So I've done this 2 different ways here. The first is fetal brain DNase peaks from the Roadmap Epigenome Project. So these would in general be marking perhaps an enhancer or a promoter. And then the second way is using genome segmentation algorithm Chrome-HMM, which includes DNase data as well as a number of other histone marks or other data to try to predict is the region in question quiescent or an enhancer or a promoter transcribed, etc. And then the second kind of filter down that we can do is say, okay, now if we look at only the elements that are active in the fetal brain in the 80% of our probands that have a neurodevelopmental disorder, we again see that the enrichment is concentrated in the probands that have a neurodevelopmental disorder and have a mutation in a fetal brain active conserved element. And while the numbers are low, we don't see any enrichment in the fetal brain active conserved elements for the patients that don't have a neurodevelopmental phenotype. So, however, I think it's important to say that the excess we see here is about 30%, and this was very borderline with about 4,000 trios. So it really took us to 8,000 trios to be clear that this was a real signal and not something that was possibly statistically spurious. So I think it's important to think these big numbers when we're looking at especially de novo mutations and non-coding elements, because it's going to be really difficult to see this subtle of an effect in less than 1,000 people, for instance. So the second thing that we wanted to do is look at any of these elements where you see more than one mutation. So this has been very successful in identifying novel developmental disorder associated genes because the chances of seeing multiple independent events is unlikely and we can actually account for this using the mutation rate model. So what we did first is look for the number of elements where we see at least 2 de novo mutations in our cohort. And we see about a 2-fold enrichment. So we observe 31 elements with 2 or more de novo mutations, and we expect to see about 15. So this is indicating that some of these elements in this pile of recurrently mutated elements are probably disease associated, but we can't exactly pin down any one of them as genome-wide significant in the way that we'd think of doing it with the genes on a Manhattan plot. So this is if you remember the Manhattan plot I showed you earlier, it's really very sad compared to the one for the genes that Jeremy published just a few months ago. And the reason for this is, there's a few reasons actually. So as you can see, we only go up close to the line here, and it's a rather small element that has 3 de novo mutations, but many of these genes up here have several dozen or more de novo mutations that's observed. And there's a few reasons that might account for the sad non-coding Manhattan plot versus the really kind of beautiful one that we see in the genes. The first is a really big one, actually, that the triplet code is very well understood. We have variant effect predictor, which tells us what is a synonymous site, what is a missense, and what is a protein truncating variant or a frame shift or a splice disruption, et cetera. And this allows us to, this gives us a lot more power in the genes. So this plot, in plots like these in general, are generated only looking at protein truncating variants or protein truncating variants and missenses. And so the denominator of this, of the observed over expected in these genes is much lower than the whole gene. The second thing is a simple one, that genes are actually longer than the elements we have. So the gene is 1,500 to 1,800 base pairs kind of median, while the non-coding elements we're looking at are 500 or 600 base pairs. So this affects our power as well. If you see 3 mutations and you expect 1, then the p-value is not going to be as strong as seeing 9 and expecting 3, for instance. And then the final thing that could explain the difference is that actually the proportion of sites that are pathogenic in an element, in a gene compared to a non-coding element, may be different. And I'm going to argue that this third point is what's driving the majority of the signal. So I'll just focus on how we kind of can account for these first 2 by empirically downsampling the data in the genes. So if we start with the 94 genes that were discovered in Jeremy's paper that was published a few months ago, and we just ignore the VEP annotation, so we say all de novo mutations we'll consider to be putatively pathogenic, including synonymous missense or protein truncating, then we lose about half of our power straight away. So only about 47 genes would be discovered, or closer to 45, if we ignored VEP in that analysis. And then finally, if we travel down this graph from right to left, if we downsample the genes to the median size of the conserved elements that we sequence in this analysis, we lose again about half of our power. But we'd still discover 15 to 20 genes if you downsample them, so you only select, say, a third of the exons and you ignore VEP. Actually, we'd still discover 15 or 20 genes. So what's left over is that, in fact, the proportion of mutations in genes might be fundamentally different than non-coding elements. So approximately 8 to 10% of mutations in a gene will cause what we'd call loss of function. So that would be frame shifting indels, protein truncating variants, or a splice disruption. And so it seems that that number very likely must be much lower in non-coding elements. So what we tried to do was, given the data that we see, which is observing 0 genome-wide significant elements in 8,000 trios, what is the probability that this would happen under across these 2 different parameters? The first being the proportion of mutations in an element that are pathogenic with a dominant mechanism, and the second being actually how many elements out of the about 2,700 fetal brain active conserved elements that we began with have a dominant disease mechanism. So from this analysis, it's clear that there's either almost 0 non-coding elements that have a dominant disease mechanism, or if there are non-coding elements that have a dominant disease mechanism, that the proportion of sites within those elements that when mutated are sufficient to cause this disorder must be less than 2%. And this has real implications on the power that we would expect to have in studies like this. So for instance, if it was about 0.08 or 8% of mutations, like what we'd expect in genes, then we'd actually have reasonable power not reasonable, but we'd have about 20% power, 20 to 25% at 6,000 or 8,000 probands. So you should start to discover some disease-associated elements. But if the true proportion of mutations that are pathogenic with a dominant mechanism is more like 2%, then our power is actually incredibly low at the number of trios we have. So this is why I said earlier that, and in fact, in either of these cases, with 1 or 2 or 3,000 trios, our power is next to nothing to predict disease-associated elements. So, did somebody have a question? No. Sorry, just hearing some talking in the back. So if we take this plot on the right of the power given a pathogenic mutation density of 0.02 and extend it out, it gives, I think, what is a really interesting picture of how the field of interpreting de novo mutations or variants in general and non-coding elements may evolve. What this basically says is that we'd have to go well over 100,000 trios, the gray plot here, to discover a good proportion of the disease-associated non-coding elements without a tool like VEP in the non-coding elements to define what is benign and what is a damaging mutation. But if we can develop a way to better understand which mutations in non-coding elements are likely to be pathogenic versus those that are likely to be benign, even at a more reasonable number of trios, 40,000, we'd get about a 16-fold increase in power. And you can also imagine on this graph, there's really a family of curves between these two. So it's not going to be a perfect predictor or no predictor, but even tools to better subset the nucleotides within a particular element as being likely to have a dominant pathogenic mechanism versus those that don't will improve power dramatically. So the last thing we wanted to do was basically take these results and, because we've only sequenced a subset of elements, try to extrapolate those genome-wide. So as I mentioned earlier, we see no excess of de novo mutations in either of these two classes of enhancers. And we see about a 30% excess in the fetal brain active conserved non-coding elements. The other thing that it's in the supplementary materials of the paper, but was a bit of a secondary analysis, is we wanted to look back at the VISTA enhancers and say, what if we focus on the ones that are conserved to the same extent as our conserved non-coding elements? And we do see some enrichment there, but it's small numbers, so it's hard to say for sure whether this will be the case for enhancers in general. But I think in the context of the data that we have from the conserved non-coding elements, it makes sense. So basically the picture that's emerging from this data is that regulatory elements that are highly evolutionarily conserved and active in the fetal brain do contribute to developmental disorders. And what we'd like to do is use the evolutionary constraint as a predictor to extrapolate this excess we see genome-wide. So if across the bottom of this plot here is you can think of it as the amount of real estate genome-wide at a given level of evolutionary conservation. So the way this was generated was take all of the high confidence DNase peaks in the fetal brain genome-wide and ask what their level of evolutionary conservation is. And so we've got about 47 megabase that's not particularly conserved at all, and a little over 5 megabase that is fairly reasonably conserved towards the top. What I've plotted above with the dots is our observed over expected de novo mutations in DDD. So as you can see, around 0.8 is where we really start to see an enrichment for de novo mutations. Although our data is a little more sparse in the center here, I think it'll be really interesting when the first large whole genome trio data set comes out to see exactly where this curve takes off. I think that's a big area of uncertainty, whether non-coding regulatory elements that are in the kind of intermediate range here will be contributing to some extent, because at the moment our extrapolation has them not contributing at all. So effectively, this allows us to take the area under the curve here based on our data in DDD and extrapolate it down to all of the real estate genome-wide. And this estimate yields an excess of about 1 to 3%. And that it would explain about 1 to 3% of the missing diagnoses in DDD. So I've only looked at DNA's hypersensitivity sites here, so this might be an underestimate. But if we look at, if we say take all of the conserved elements genome-wide and try to predict function for them using Chrome-HMM, some slightly more liberal cutoff, we still only really get to about 5% at best. So I think it's fair to say that mutations in single nucleotide mutations in these regulatory elements should contribute to a minority of these cases, but still a non-trivial set of people. This would be 40 to 140 families in this case. And so the other thing I think to note is that we cover the high end of this evolutionary conservation spectrum within our capture set to a decent degree. So we get more than half of the elements genome-wide that are in the 0.9 to 1 range, and about a quarter of the ones in the 0.8 to 0.9 range, but actually very few that are less than 0.8. So that's why I was saying earlier, there's some uncertainty over what elements that are a little lower on the evolutionary conservation spectrum.
So do you expect that this type of non-coding mutation might result in a less severe type of phenotype? Did you look at the impact of endophenotypes in the individuals?
That, I think it's a great thing to look at. We didn't specifically look at severity of the phenotype because it's not. We found that the clinical phenotyping data is it's not particularly quantitative. It's a description based on the human phenotype ontology. So it's really actually difficult to judge the severity. I think it's also important to note that when we have a 30% enrichment, say, or a twofold enrichment in the recurrently mutated elements, that we're looking at a mixture of false positives and false negatives. To answer your question, we haven't looked at severity, but I think it certainly would make sense biologically if these mutations are just modulating the expression of a gene rather than taking it down from 100% to 50%, say, like a protein truncating variant.
Right, because you can also think that the trio design per se might not be the right design for looking at these elements, and maybe looking at ultra rare variants in case control might add additional signal in this region, because the frequency for this element is probably going to be slightly higher because they're tolerated.
So I think it is good. We made a slight attempt at looking into rare variants by looking at overtransmission from the parents, but I suspect that it's probably an underpowered analysis in this case. I think without the ability to discriminate individual nucleotide changes and what they do within an element, it's always going to be hard to separate the signal from the noise in these elements, because so many of the variants in these elements we should expect to be benign, and unless we can separate those out, it will add noise to a case control analysis as well, I think.
Did you try to play with any of the meta-analytic tools like CAD, EIGEN, and these other tools? Did they help in any way?
Yes, we did. So I looked at CAD, Fathom MKL, and Genomizer. So CAD, if you check out the paper, I put the CAD figure in figure 1 and Fathom MKL and Genome-wide in the supplementary materials. What we found is within these conserved elements that all three tools can to some extent separate benign from possibly damaging, but they don't have any resolution to detect highly damaging variants if they're present.
Okay.
So for a CAD score, anything above 10, 12, 13, 14, all the way up to 20 is all, from a purifying selection perspective, under the same level of purifying selection. Whereas if you did it in coding regions, there's this very clear gradient where every step up with a CAD score corresponds to stronger purifying selection.
Yeah, I guess also a question is since CAD include fast cons and these other conservation, are they actually adding any information above what, or they're just capturing elements that are simply more conserved?
Yeah, so I think that's one. In CAD's case, it's probably recognizing that it's an ultra conserved element and giving almost every variant in the element a similar score. I have found that the philo-P score, so the nucleotide level conservation, does possibly provide a little more information than CAD, and in the context of a highly conserved element, that a highly conserved nucleotide is under stronger selection than a nucleotide that is not highly evolutionarily conserved. So I think I guess the answer is there's some information in each of these tools. It's just figuring out how they work in the context of a specific set of elements. Like the conserved elements, as you've said, are so conserved to begin with that tools like CAD that already have that in their algorithm might tend to call everything as being damaging.
Okay, thank you.
Yeah, just as a comment from my side, with regard to Andreas' commentary, it's I mean, since you include also like chromatin information, and chromatin is, over time interesting, right? So if you have the fetal chromatin signatures, but you can also look at infantile signatures. These neurodevelopmental disorders have all different times of timing, and usually they're before childhood, but there's a range, right?
Yes, that's right.
In the future, this might be interesting if you see specific chromatin signatures for specific variants, and if you can correlate it with age of onset. In the future. Now it's highly underpowered, but who knows?
Yes, that's right. I think there is definitely, when we have more whole genome data, a really cool kind of tissue-specific, multi-level, hierarchical way of looking at this, because we have time ordering of development, and there might be a kind of tissue-specific way of breaking down when these elements are active, and if there's some there's some critical window in which a particular element is active, for instance, that might shed more light. But I think you're right as well, that we've only looked at a specific time point of the fetal brain, but where these mutations are having their effect could be anywhere in a long string of developmental time. Right. So we cover a good chunk of the ultra conserved elements within this set of 4,000, but I think it remains to be seen exactly how this curve shapes out as more data is added across the whole genome. So just to summarize quickly, the first thing is that when we look in the exome negative probands, those are the ones without a diagnostic mutation in a protein coding gene, we see a 30% excess in the fetal brain active conserved elements, which accounts for approximately 1 to 3% of the missing diagnoses in this cohort. While we can detect a signal for recurrently mutated elements, there's no individual element that's genome-wide significant, which is, I think, a really valuable piece of information that led us to hypothesize that probably a very small proportion of the mutations in these elements are sufficient when mutated to cause a developmental disorder with a dominant mechanism. And to further tease these out, we need better tools to predict the effect of individual changes in non-coding elements. I think that's probably very obvious to a lot of us in the who work with these kind of tools, that they do a good job, but there's still much to be learned, whether from an experimental perspective using massively parallel reporter assays, or from a machine learning and bioinformatics perspective, figuring out how to take these elements and figure out what exactly are the critical regions. And I think large whole genome data sets like Nomad, for instance, will really be helpful, much like some people are doing with missense mutations or loss of function mutations, figuring out which sections of the genome are missing variation, and are there particular nucleotide level patterns that are predictive of purifying selection. These elements will really go a long way towards boosting power in these studies. So I'd love to take any questions or feedback that you all have on this. I think I still definitely consider it a work in progress because there's a lot still to be done. I'd like to thank first and foremost the families and patients in the DDD study. They're very patient and don't always get a diagnosis, and I think that's the number one goal of this project, is to try to get as many diagnoses as possible. But it also allows us to do interesting science like this. And also my supervisor, Matt Hurls, as well as all the members of the DDD team and other members of the Hurls group that have contributed to this analysis. So if you have any questions, or feedback or ideas, I'd love to take that now.
Thank you very much. This is certainly a very good presentation and also a topic anyway. Are there any more questions?
So is there any reason to believe, you look only on those that you call exome negative,